Well, thank you so much. Hi, everyone. I'm very glad to be here today at the site of Summit to present what we think is the next leap in multi-omic data analysis. And so I'm going to talk to you today about the way we are unifying multi-omic data sets with biomics intelligence. And biomics intelligence is the result of the partnership that SurgeCare is carrying out with Standard BioTools to analyze multi-omic data sets. And so I want to start by showing you something that we all know very well, which is that we now have unprecedented access to millions of data points. And I want to say that Surge is actually a spin-off out of Stanford University, out of the Goodyear lab that is interested in looking at multi-omic data sets. So really integrating those millions of data points, millions of signals on different very rich clinical outcomes. And those can be surgery, pregnancy, COVID, stroke, and so on and so forth. And the big problem that we have there is that usually in those studies, we're interested in modeling those outcomes, but we don't necessarily have a lot of samples from a lot of patients. So we're in this problem where we have those millions of data points, but not necessarily enough samples to use classic methodological tools. And so technically, and the results of this is that only a handful have progressed, a handful of those studies have progressed to an actual clinical impact. And so I want to emphasize that the result and the actually cause of this is probably that omics data sets are super difficult to analyze. The complexity of omic data sets bring the translation to clinical application actually is very difficult. And I want to take an example that we've been developing at Surge, which is on prediction of surgical complication. We've been carrying out, as an example here, a big clinical trial where we collected whole blood and plasma in surgical patients. It's on more than 280 surgical patients undergoing major abdominal surgeries. And our goal was to identify an immune signature capable of predicting surgical site infections and more generally different types of complications. We had up to 10 different clinical outcomes in this study. And so for this, we're able to derive a big data set of, in this situation, 3 different stimulations for the whole blood and more than 1,000 proteomic expression and a proteomic plasma assay. And we wanted to integrate this huge data set with 300 million single cell events, then and more than 1,000 features for the bulk proteomics, with this one goal to identify the signature to predict surgical site infections. And so we're in this problem here where we're deriving actually with gating more than 1,300 immune cell features, more than 1,000 proteins, but we only have 283 patients. And so we're limited. But in this case, we're already in the case where we have a lot of patients in this clinical trial. We've integrated patients from 5 centers that we've been recruiting for more than a year. And what we see with this setting of study is that we're in the case where classic univariate statistical analysis are not adapted to give us an answer on this data set. It doesn't give us really clearly where the signal lies because it will only look at features one by one instead of looking at the capacity to integrate those multi-omic data sets together and look at a multivariable signature. And so we've been interested in developing the frameworks that will allow us to translate into the clinic and that have 3 optimal properties, which is prediction performance. And here the question is, can I have a signature that is actually predictive of my outcome? Here, can I predict surgical site infections from this immune signature? The second question is, can we find a signature that is also sparse, meaning that it's finding a limited number of biomarkers that can translate into the clinic to have a good predictive performance still? And 3, are the features that we're measuring actually relevant for our clinical outcome? So even if we find 3, 4 biomarkers, are they all linked to the clinical outcome that we're looking at, or is one of them a spurious signal? And so based on this, we've been developing that framework that is called Stable and made the cover of Nature Biotechnology in last October that is actually precisely doing this. What we're doing is that we're putting in noise, adding noise in our data sets that is mimicking the signal that is non-informative in the thousands of potential features that you have accessible. And we're using this to discriminate between what is actual signal from what is just behaving like random noise that we put in a data set that mimics the omics. And so here is what this algorithm actually does. It tailors the signal over noise ratio to each of the different omic type. And so for our study, we have two different omic types. One is cytof and one is proteomics. And on cytof, you can see here on the graph a representation of all of the features that you have available. So each line that you can see, whether it's black or it's in color, is a line that represents a feature. And we're comparing here the selection of those features, the robustness of those features in comparison to noise that is inputted and that is shown in dotted lines here. And what we can see is that this pipeline allows to basically adapt the threshold automatically without a human bias to the signal over noise ratio of your data set. And now you're able to select the relevant features from your data set and translate them into a clinical application. I want to emphasize that we are able to also translate those on imaging technologies, and they're actually super adapted to our pipeline because those technologies like IMC data are high dimensional, multimodal, and so they have a high potential for translation here. We're able to derive features that are relevant, biologically meaningful, and integrate them into the pipeline to then extract what's relevant from what's noise on your clinical outcome. And so this is what we've built at Surge around this pipeline and integrating things that have been already developed. Three main things. One is the capacity to derive biomarker selection using our pipeline, but also different type of machine learning algorithms. So we have a robust evaluation framework. We're evaluating in cross-validation and when we have available validation. We build this automatic machine learning framework that allows to select the best model and best input in the pipeline, again, without human bias, to derive what will be the best features in your data set to derive for a clinical application. And then we're able to integrate the multi-omic data sets to give you what is actually the optimized signature for your own clinical application. And so going back to the surgery setting here, what we've been able to do is train the model on the first 150 patients, find 6 relevant biomarkers, 5 from the single cell data set, 1 from the proteomic data set, and integrate them into signature that is predictive on the validation cohort. And what we've shown is an area under the curve of 77%, a predictive value, negative predictive value of 95%. And what's really interesting here for us is that it's now not just a study. We've integrated this into a product that is approved to be used in medical settings, and it's called PRECYTE. And the goal of this product is to be able to tell which patients are at risk of developing the surgical site infection after major abdominal surgery. And so we went from this big data set of more than 1,600 samples, 500 unique single cells, used AI to find 6 relevant biomarkers that are now integrated into a product that is used by patients and transforming surgical care patients in actually with the first pilot in UPenn, and we're collaborating with Immune Health to make this possible. And so we've shown that we're able to go from the multi-omics to actually changing the clinic with this specific test. And so this was all made possible by our platform that is called biomics, and that is the reliable AI solution for multi-omic biomarker discovery. And so I'm very glad to be here today to present this and make this available also for Standard BioTools customer. And we make this pipeline and this platform available through the services or directly in collaboration with academia to help everyone that generates multi-omic data set translate their application on clinical outcomes to find reliable biomarkers. And so diving in what we do with biomics, we're able to deliver scientific insights within days. We build the platform so all the biomarkers and the results are interpretable. Everything is linked to a statistical meaning that we want to minimize false discovery rate. We want to link the discoveries to actual biomarkers that you can understand. They're reliable. They're built on machine learning pipelines that have been published in peer-reviewed journals like Stable. They're fast. We can perform this and give reports within days. and they can be performed also on small cohorts, so starting with as low as 30 patients to link this to clinical outcomes. It is the missing link with what has been already available with the services at Standard BioTools, so being able to go now from really a one-stop shop from panel design, acquisition and analysis, and now we're able to directly plug this into biomics for advanced AI analysis. And we're able to do automated pre-processing if needed, or directly take the pre-processed data sets so that we can integrate this into our multi-omic pipeline and link this to the clinical outcome to give a clear scientific report of what the biomarkers are linked to your outcome and their robustness for translation in clinical application. I want to just showcase an example of an automated pre-processing that we're able to give out. What we do is that we have an efficient cell gating feature extraction for CyTOF and IMC when this is needed, again, for teams that have no expert or no expertise in deriving features from single-cell data sets or IMC data sets. Those are very reproducible, reliable, and built on prior knowledge to extract information on known cell types. And we're integrating this into a platform that allows to extract the relevant features, spit out a report with the relevant biomarkers, and give this fast, reproducible, interpretable biomarker discovery phase. It's adaptable to all of the study designs. It can be modeled on continuous outcomes, so survival analysis, for example, prediction of future events, multi-class outcomes like disease stages, or binary outcomes like our case in prediction of surgical site infection. And so now this pipeline is available through the services. We're also happy to collaborate directly with labs or core facilities, but I'm also excited to tell you a bit more about what we're currently developing and what's coming up next. I think one of the things we're very excited and happy about at Surge is that there are more and more papers and science about the capacity to use foundation models to translate the thousands of data sets that have been already acquired on many different use cases and try to use this to make future studies more efficient and extract biomarkers in a more efficient manner. And so this is a perspective article that was published in Nature late April this year that is actually telling how we can use those new technologies, foundation model technologies, to integrate omic data sets and push this for new discoveries and help actually translate the new omic technologies to new findings that will be accurate even with small sample sizes. And what we are building today is the first foundation model for the reliable biomarker discovery. We are collecting data sets from multi-omic data sets and integrating this into a foundation model for multi-omic data sets. And so what this will help us do is have a model, AI model, that, so to say, speaks the language of multi-omics, speaks the language of biology, and is able to derive relevant insight and features from the pre-trained model that have been set on thousands of potential data sets. And from what it gives us, which is feature extraction, easier than what was done before, in a more stable way than what was done before, we'll be able to translate this into precision medicine tools, meaning biomarker signatures that can really impact the clinic, which is our goal. And so to sum up, our mission is to help researchers be empowered to change the paradigm and move from the trial and error medicine that is considered that we have to measure one parameter at a time. We're going from symptom-based medicine and kind of agnostic to this one-size-fits-all treatment paradigm to a personalized medicine where we're measuring all those biomarkers, and we are capable of using that information to derive relevant biomarker signatures and translate those biomarker signatures into the clinic. And so we're already collaborating with a lot of academic institutions, helping them translate their omic biomarker study into finding where the relevant biomarker are and actually helping them translating this to the clinic. And so we'd be happy to talk to you to collaborate further. And thank you so much. I'm happy to take any question.